list of things. We've been talking about nutrients and waste products, and gases are not there, but gases are going to be there. And hormones we touched on a little bit, like kind of mentioning, even though we haven't done anything yet officially. And electrolytes, we've talked about these items, right? We focused on them maybe working in the nervous system, but they're going to be floating around. And then recently we mentioned some of these plasma proteins, right? When we were talking about osmotic pressures and having these proteins suspended in the blood that are just there, basically, like their main thing is to take up space, right? And contribute to that osmolarity. So our list of plasma proteins, albumins, globulins, which include immunoglobulins, which we'll see in the immune system. And then fibrinogen, which is an item that we will talk about today. It's going to factor into this whole clotting business. So we'll see that a little bit later. But all of those items all together, they're all proteins, and they're all in the category of plasma proteins. So again, all that stuff should have been, kind of mentioned in anatomy, but here's where we're going to really talk about them a lot today. And then we've got blood cells, and that's coming up in the next slide. Also should be some anatomy review. And I just put up here, if you hear the word serum, when we're referring to blood, is the plasma part of blood with the fibrinogen removed. So that would be serum, if you happen to hear that, right, in the future, going out there, and someone asks for that or something like that. So, let's see. Good. So here's our review of blood cells in the blood, obviously. We're going to start with a stem cell, and our fancy word for a stem cell that could turn into any one of the blood cells would be hematopoietic. Hematopoietic. You might also hear hematopoietic, and that's also fine. Either one. So the word that you see in front of that word, hematopoietic, is pluripotent. Pluri is like plural, right? And potent, what does that word mean? Just like, maybe not here in physiology, but potent, what does that mean? Strong, right? Potent. So, so something that is strong and having to do with plural or many, right? So what this word refers to is a single cell that has this, let's say power, right? It's like a superpower. To be able to possibly turn into one of many things, right? So you see with the arrows, we start out with one. You could go in this direction, the lymphoid stem cell direction, and then ultimately become a lymphocyte. What we're remembering from anatomy are types of lymphocytes. Basically, we had two basic categories. Anybody remember? Lymphocytes. There were letters. There was something lymphocyte and something lymphocyte, and they were letters. T and B, right? Okay, T and B. We'll talk more about those in the immune system chapter, right? But that's just a little, put it into the back of your head there. We could also go in this direction, myeloid direction, and then that cell can then take any one of these other pathways and become perhaps a red blood cell or erythrocyte, or perhaps any one of these white blood cells. Or this item down here, megakaryocyte, mega for huge, right? And you can see this is huge compared to all the other ones. And the megakaryocyte is in charge of breaking off little bits of itself, and those little bits are our platelets. And we'll see how we use those a little bit later as well. Good? So here are all of our choices. And you definitely want to know all of these, right? All right, because that's pretty, that's kind of your baseline. Here's a table. This actually comes from the Merritt Anatomy book that shows the different cell types that were on the previous slide. And so you see the names and the picture, and those all match. And then you see more details about each one. Don't worry about all the details. Let's see. Yeah, don't worry about all these other details. Just be sure that you know the names and the pictures, right? And that's just looking at the previous slide in a different way. Good? All right. So this should be a review of a red blood cell or erythrocyte, review from anatomy, so you should already know the shape and why it has this shape, and you should already know what is packed inside of these, hemoglobin, right? So this should also be a review from anatomy. So we've got two pairs of globin protein chains put together, right, attached together. What type, let's see, is the answer there? No, good. What type of protein structure or what level of protein structure is this whole thing? Good. Woo, now you're going to pause. Quaternary. Woohoo! Very nice, because we've taken, in this case, 4 separate chains. I mean, it didn't have to be 4, but for hemoglobin, we take 4 separate chains. Each chain has its tertiary structure, and then we put them all together, and that gives us quaternary structure. Remember that? From, oh my goodness, way back when, right? Oh, wow, super awesome. And we've got a heme molecule associated with each of those chains. So we have 4 heme molecules per hemoglobin molecule. And this heme molecule has a ring shape to it and iron. An iron ion of +2, don't want to be crying on that one, hangs out in the center of that there, ready to attract an oxygen molecule. The oxygen molecule will not bind to the iron ion, but it will like to be near it, right? So one hemoglobin molecule can carry 4 oxygen molecules, good? By having the oxygen molecules hang around with the iron ion. Good. So this also should be an anatomy review, but we're going to add some details for physiology. So you definitely want to know this recycling process of red blood cells. So let's start at the top. We're going to make the red blood cells in red bone marrow, and we see, remembering from anatomy, they're showing us the end of a long bone, right? And in an adult, it would be the ends of long bones, and we had another location, right? Like the ilium and iliac crest specifically as a good location for red bone marrow. But that's in adults where blood cells are going to be made. So we see they're showing us that, and they will get released into the bloodstream, circulate around, 120 days, and then they're done, right? And let's remember back to anatomy also, I hope so. Why did they really only last 120 days? It was kind of a big reason, and it was because of how they were built and a particular something that they're missing. Nucleus, right, so they're missing a nucleus, good? So without a nucleus, what can you not do? Right, you can't reproduce, right? So eventually you're just going to like wear out and then like that's it, you're done, right? So that's what's going to happen. And when the red blood cells reach that point, the liver and the spleen are in charge of recognizing that sort of worn out state of a red blood cell, and they will sequester the old red blood cells and break them down. So they break them down, and we're just going to focus here on what are we going to do with this heme and this iron, right? So heme, right, our ring is going to be converted. I don't know, can you guys see this in the back? I'll make it bigger if you want. Let's make that bigger. Let's see. Oops. There we go. Oh, is that any better? I don't know. All right, I think is that a little better? It's a little better. All right, so heme is going to be converted to some item related to this original ring called biliverdin, and then that's kind of an intermediate thing. And then that biliverdin is going to be converted ultimately to bilirubin. Some people have heard of bilirubin coming in to class, but if not, that's okay. We're going to learn it now, right? So we're going to end up with this molecule. It's called bilirubin, and bilirubin ends up being secreted by that liver in bile. And we probably remember a little something, I don't know, about bile from anatomy, right? A little bit. We'll talk about it more. Bile and bile salts, right, and that role in digestion, right, the digestive chapter. So for now, we're mentioning it. We'll talk more, right, in the digestive chapter. All right, so far so good. So that's what we're going to do with this. Get rid of that, and then we've got our iron. Remember we had the 4 iron ions. I think maybe to be slightly more correct, if they're going to put the 4 iron ions in the picture, I think they should have put the 4 heme groups. You know what I'm saying? The heme molecules there to, you know what I mean? Because I think this is a little bit misleading or something, or not quite right. So either have 1 and 1 or 4 and 4, right? So just saying that will be more correct. We'll rewrite the book. So anyway, more correct. But here we've got our 4 iron ions that have been removed from the heme, right? And this one over there to end up in the bile. And then this is going to hang out here. We can't have iron sort of floating about in the body, just sort of, I don't know, like just nakedly floating around the body. So we're going to bind it to a protein that will make it safe to float around, right? So the name of the protein is transferrin, and maybe a way to remember that is like transferring, right? Like carrying the iron, maybe that's a way to remember. Though the truth is this root F-E-R-R is referring to iron, right? If you remember from chemistry, right? So transferring is just sort of a little trick to remember, but keep in mind it's F-E-R-R referring to iron, right? And it's trans is more of like the carrying part of it. All right, just, I don't know, just seeing if that'll help you remember stuff. So we're going to carry the iron using this protein called transferrin. So we can carry that through the blood and keep the body kind of safe from this sort of naked iron. And then we can move the iron from transferrin to yet another protein and store it, right? Let's say we have some extra, right? We can store it in the liver using this protein, ferritin. Ferritin, and again, you see that F-E-R-R for iron. Good. So transferrin for, let's say, transferring iron, right? Can float around in the bloodstream, and then ferritin for storing. And we're going to store it in the liver. I don't know if anybody can come up with like a little trick for remembering the word ferritin having to do with storage. I don't know.
You're putting it in the liver.
In. You're putting it in. I like that.
What about you putting FE? Because FE is essential iron.
Yeah.
So FE.
Yeah.
So storage is iron.
But I kind of like the it in, like fair it in, like you put it in.
Iron
It's, yeah, something about putting it in, or putting iron in. That works. That works. All right, maybe. If that works for you, good. If not, you know, I don't know. Just saying, because they're so close to each other, and you know I'm going for it, right? You know that's the thing. I'm gonna put the question, right? So, all right. Eventually, we're going to see there's going to be a way to get rid of iron if we have too much iron in the body. It's not good to have too much iron in the body. So one way to get rid of iron in the body is to bind it to that ferritin, but instead of storing it in the liver, we can do that in the small intestine. We'll see this, like I said, in the digestive system, but in these epithelial cells in the small intestine, and then just allow that. We should know from anatomy that epithelium replenishes itself, right? Replenishes itself like every once a week at least. So when the old cells just slough off, if we've got iron bound to ferritin inside that cell, and then the cell just leaves and ends up in the feces, out, right? Make sense? All right, so that's a way of getting rid of excess. Good. So we're going to see that again. We'll revisit that. What else? And then you just kind of recycle everything you've broken down, right? We do have to take in iron in the diet. Let's see, I think that's on this side. Yeah, slide. Okay. So over up here and over here, we do have to take iron in the diet because of the iron that we lose, and women lose more because of menstruation. We've got sloughing off of the lining of the uterus, so we've got like double sloughing, right? And so women have to take in more iron because we need the iron, right, to build the blood cells, right, to finish off that hemoglobin. So other things that we require in the process of building red blood cells: 2 vitamins. So we need the iron, right, for the hemoglobin. And to kind of build the whole cell, we need 2 additional vitamins. One is folic acid. So we might have to remember, I should have put the reminder slide. Remember our list. Oh my gosh. Which chapter was that originally from? I'm trying to remember. Is it like the enzyme chapter? Remember we had the list of vitamins? I'll find it, like maybe during the break, remind me. But the original list, remember the original list of vitamins. And so far one of the vitamins that we have for sure learned. Let's see.
B vitamin.
You have to remember.
B.
What?
B.
Huh?
I think it was B vitamin.
Now you can see. What have we learned so far of vitamins? Like, and we've used them, like, we did some in metabolism, and they were like coenzymes. There were some B's.
Yeah.
Oh, there was riboflavin, niacin,
Pantothenic.
woo, pantothenic, right? So those were our 3 biggies. They happen to be all B vitamins. And we learned about them in the metabolism chapter, right? Okay, so far so good. So we're adding to the list of vitamins. Oh, I think we also talked about
Vitamin A.
Yes. Remember we were talking about the eye, right?
Yeah.
What did we talk about?
A.
A, right? Okay, good. So that's 4 vitamins that for sure we've had like detailed conversations about. So now we're going to add to our list. So here we're adding folic acid, and we need folic acid to build. Ooh, this goes back. Remember thymine? Oh my gosh, like nucleotide bases, right? And all of that. Oh my goodness, that goes back. And then B12, which kind of helps folic acid. Good, to do its job. So we need these 2 items to sort of complete our big process of making the red blood cells. Good? All right. Building the vitamin list. Good. So, let's take a look at this flow chart, which is talking about how do we regulate or control this process of making red blood cells. The fancy name for making red blood cells is erythropoiesis. Erythropoiesis, making or building red blood cells. All right. Because we always have to make new ones, right? If they're dying off every 120 days, right? Or like the one is made and then 120 days it's done, and then you've got some that are overlapping and all that. But every 120 days, and a group of red blood cells are dying, so or being recycled or something like that. So we do have to make new ones to replace those, and we should have control over that, right? Because just in case, I don't know, you have a moment where it's like, oh, whoa, well, we've got enough, right? You don't want to keep making more, because then the blood is going to become too full, right, of red blood cells, it'll become too thick. And remember, we talked about viscosity, right? It was on one of those slides. Oh, it was that big slide, right? It was the big slide. And it was kind of over here. Remember, there was viscosity and hematocrit right down here. So you don't want that to go up. It's too thick, right? You also don't want it to go down and then not enough, and then the body's not kicking in, right, to make more. So we should have control so that if you have too many red blood cells, whoa, slow down, or if you have not enough, okay, hurry up and step it up, right? Let's make more at a faster rate, right? So the kidneys are going to be responsible for detecting specifically the oxygen level in the blood, the oxygen that's being carried, right, by all of that hemoglobin that's packed in all those red blood cells. So far makes sense? All right, so that's one of kidneys' jobs. We'll see, of course, many, many more jobs when we get to the renal chapter, but let's introduce this one job here in this chapter. So, kidneys are going to sense, right, oxygen levels as the blood passes through the kidneys. And let's say they're giving us an example in this flow chart that the level of oxygen is low, right? Low. All right, fine. So the kidneys will sense that, and they will start producing and secreting a hormone. Here's the name of the hormone, erythropoietin. That's a good name because it has erythropoiesis, right, in the name of the hormone. So it tells you what it does, right? It's in charge of helping with making red blood cells. So kidneys are going to produce and secrete more of this erythropoietin. So more of this erythropoietin is going to float around in the bloodstream because our definition of any hormone is something secreted in the blood and then it travels and goes to a target, right? Goes to a target organ. Our target is going to be the bone marrow. That makes sense because that's where we're going to make these things, right? So our target is the bone marrow. And the bone marrow is going to receive that erythropoietin. Erythropoietin is going to signal bone marrow, go, right? We need some more red blood cells because we're not getting enough oxygen out here. And maybe if we have more red blood cells, we could grab more oxygen from the air, which is respiratory, and then deliver it, right, to the tissues and have that sensed by the kidneys. All right, so bone marrow is going to respond by stepping up that production of red blood cells. Great. And then we release more red blood cells into the bloodstream. And we can, in fact, pick up more oxygen at the lungs because we just have more cells, right? We have more red blood cells. And kidneys detect that. Good? Kidneys detect that. Now we're back to normal, and we're happy. Good? All right, so I think that's pretty straightforward. Let's see. Having said all that, anemia is our word for basically not having enough red blood cells, right? Or your red blood cell count is on the low side. So we've got some causes. As always, I'm not going to test you on pathology, right? But let's just kind of, go through them because sometimes, like I always say also, when we look at an abnormal situation, sometimes it helps us to understand normal. So we could have not enough iron being taken in the diet. That makes sense, right? We could also have not enough of the two vitamins that we needed, right, to also help with erythropoiesis. Make sense? Right? If we don't have enough of the building blocks, we just can't make enough red blood cells, and there's our definition of anemia right there. Okay, so that's a couple of causes there. We could have a problem in the bone marrow, and maybe the problem is a cancer in the bone marrow, or maybe the problem is a drug that maybe one of the side effects is to dampen that bone marrow's actions. All right, so there's that. Sometimes the cancer drugs are the ones that do that. Wow. All right. So anyway, but that's a whole big topic. Let's see. We've mentioned, when we were talking about blood pressure, remember we mentioned hemorrhage? Like big bleeding? So that's obviously, right, if you just bleed out, you've lost a bunch of red blood cells. Okay. There we go. That's one. Wow. If the kidneys are not producing and or secreting enough of the erythropoietin, right? Oops, that could be a problem, right? Because then the bone marrow is not getting the signal. Make sense? To produce more red blood cells, so you're staying on the low side. Okay, so there's another one. And then, if the red blood cells are being destroyed, and they're giving us an example of sickle cell disease when the red blood cells have an abnormal shape, and then they get sequestered by the spleen and destroyed, and you just keep making all the wrong shaped ones, and then you end up with low, right, red blood cell count. Okay, so these are just examples, but it helps us, by looking at these examples of pathology, it helps us to think about, like to review, oh, this is how it's supposed to go. Good? All right. Let's see. So, we've talked about hemorrhage, right? Bleeding. Here's our, I guess, sort of opposite word. Hemostasis would be stopping the bleeding. Stopping the bleeding. So hemostasis. We've got built into the body sort of 3 steps. 3 big major steps with many steps, of course, within each step for stopping bleeding. So here are our steps. Constriction, specifically vasoconstriction. We know all about vasoconstriction, right? We're good on vasoconstriction, right? We're going to add just a little bit of the local stuff on vasoconstriction and a little bit of vasodilation again. So we've got that. And then we've got forming something called a platelet plug. We'll take a look. I think we're like 1 or 2 slides away. It's pretty close. And then after that, we've got making an actual blood clot. And we've got some things to go with that. So, let's see. I guess it looks like we're going to start with forming the platelet plug, but at the same time as talking about forming the platelet plug, we're also going to add some of the local controls to vasoconstriction and vasodilation. Remember we mentioned those in the blood vessel section, blood vessel, blood pressure section, and we mentioned the local controls. It was like nitric oxide. Remember we had our little like official intro to nitric oxide. So now we're going to add just a little more information to that information, that basic information. So we're going to kind of talk about that and the platelet plug at the same time, because they sort of happen at the same time. So, begin as usual. Vessel damage, right? You get a cut or something, right? Scrape, whatever. And the blood vessel is cut. So what happens is when the blood vessel is cut, that endothelium is cut, right? And remember, what did we make the endothelium with? Like, it was an epithelium made of what type of epithelium? Squamous, how many layers?
1.
1. So what was our word for
Simple.
1? Simple, right? So we have a simple squamous epithelium. We call it endothelium as our lining, let's say for capillaries. Let's just focus on capillaries for now. Good? All right, so we had that endothelium, let's say it gets cut, right, or ripped or something like that. Damaged, whatever. What is underneath I guess in a capillary it would be like outside, right, outside of that endothelium. What did we have to have, I don't know, to complete the structure? Like to support the endothelium. Some kind of connective tissue, right? We always have to have a connective tissue, right? Sort of by definition, anatomy. Anatomy should have been all about combining epithelia and connective tissues, right? To create things, to build things, right? Really, if you had to summarize anatomy, right? Probably at least half of the summary, maybe 2/3, would be which epithelium do you pick and which connective tissue do you pick to build such and such structure, to build a stomach, to build a capillary, to build a small intestine, right? To build whatever. Probably at least half to 2/3 of that course, right? Was basically just put it together and build. All right. So, if we have cut or ripped, somehow damaged the epithelium, what's exposed? The connective tissue underneath. So, what do we have in connective tissue, specifically areolar connective tissue, as far as proteins, right? We had structural proteins, not structural proteins inside of cells, but structural proteins that are spit out of cells and they hang around in the connective tissues. Collagen, good. Wow, that's good. You pulled that out from Winter. Good. That's impressive. All right, so collagen, right? Collagen. Oh, good. So we've got collagen. I mean, we had other things. What were, just to do the review, what were our two other types of extracellular proteins? So we had collagen. Two others. It's just to review it. What's the one that like stretches and then comes back? Elastin, right, or elastic fibers?
Isn't it adipose?
Ooh, reticular fibers. Reticular fibers. Yeah, adipose tissue was one of our connective tissues, right? Remember we had like loose areolar connective tissue, adipose tissue was in that same category, right? And with each of these connective tissues, we can choose some collagen, some elastic fibers, some reticular fibers, right? And put them in, build in different amounts, right? Okay, good. Well, anatomy review. So let's see. Seriously, I wish it would be like just A and P course, always, right? Because it's so hard to have to remember the anatomy. Anyway, that's all right. Let's see. That's what all the other schools want. Like all the schools that you guys are applying for, like that's why they set it up like this, because that's what they want, is like separate anatomy course, separate physiology course. Oh well. Bummer. But anyway, that's okay. We've damaged the endothelium, we've exposed the collagen, and the blood is like passing through, right, and is sort of sensing, right, that collagen. So we've got a protein, right, a plasma protein. Remember our list of plasma proteins? So one of our plasma proteins is called von Willebrand factor, and we abbreviate that with the lowercase v and then capital W and capital F. So that's just the name, right, of this particular plasma protein produced by both platelets and the endothelial cells themselves. Endothelial cells, it's going to turn out, are extremely busy. They do kind of a lot of stuff. And this is one of the things that they do. So we've got this von Willebrand factor floating around at all times, basically always ready in all locations to sense such exposed collagen, right? Okay, so that's good. Okay, so we sense some exposed collagen in this location where we cut ourselves, and it's going to attach, right? It's going to attach, and then the platelets are going to attach to that, right? The von Willebrand factor. Make sense? So von Willebrand factor attaches, and then platelets attach to that, right? All right, so far so good. Let's see. Once those platelets have attached to the von Willebrand, stop talking while I'm talking, ladies. Stop. It's annoying. Just stop it. Next time you're out. So, once the platelets have started accumulating on this von Willebrand factor, they are going to be activated in turn to release a whole bunch of stuff. They've been carrying around these vesicles with a whole bunch of basically chemicals, right? Chemicals that are going to trigger a whole bunch of, like a series of things to happen. So, when the platelets start aggregating, right, that's going to be our official word, aggregating, they start getting triggered to release these chemicals from those vesicles that are already there in the platelets. So, we're releasing some chemicals. I put parentheses around here because in our book they wrote chemical mediators, which is a very general, I don't know, general description, and I thought, I understand why they picked general, because if we go into details, then it becomes a true nightmare. The other side of that, I don't want you guys to just memorize like a really general word either. That seems kind of lame. So I put that in parentheses. In favor of, we're going to acknowledge, okay, that's there, right? We know there are going to be a whole bunch of chemicals released. But I thought, let's focus on this because we are specifically going to talk about this one particular item that is released. It's called thromboxane A2. We'll abbreviate that. The abbreviation isn't here, but it's on like the next slide. Capital T, capital X, capital A is a subscript 2. You see it like, I think it's the next slide. Thromboxane A2 or TXA2. So that's just one of the specific things that those platelets are going to release among all these other things. So let's focus on that. And we'll see the picture coming up. So this TXA2 is going to cause the blood vessels, specifically the smooth muscle in the blood vessels to contract. And that is our definition of what? Contracting the smooth muscle in the blood vessel wall. Vasoconstriction, right? We totally know that. Okay, and there it is, vasoconstriction. Pretty good so far? All right, and that was first on our list of ways that we can achieve hemostasis, right? Stop bleeding, basically, right? Vasoconstriction. Does that kind of make sense, vasoconstriction? Like sort of slowing down the blood flow to a location that has been injured? So then you don't, right, spurt the blood out, you just kind of slower. Good? All right, so that makes sense. So far, so good. That's kind of good. Let's take a look at this little dotted line here with the plus sign there. This is our designation for a positive feedback loop, right? Positive feedback loop, which means the more thromboxane A2 that is released and starts working, kind of cycles back and it encourages even more to be released. Make sense? All right, so that's our positive feedback loop there, so that we can, right, vasoconstrict more. Right? Okay, so that makes sense. And then now we've got this side thing, the formation of the actual platelet plug. That's going to be, I think it's next, our picture. Platelet plug. Yes. All right, so here's our picture of a platelet plug. So we've got our capillary with our endothelium and these blue squiggly lines representing collagen in the connective tissue outside of that. And here's our, right, the cut where the collagen was exposed and the blood sensing that collagen. And I guess they didn't show, like they didn't make any sort of shape or anything to represent von Willebrand factor, but we know that that would have bound, let's say maybe, I don't know, like right here at sort of the base of the platelet plug, like we know that that would be there. When we rewrite the book, we'll add it. So we'll add that in. And these blue, they're sort of spheres. I don't know, they're like ovoid spheres. But these blue things that look like little pills, those are representing our platelets. And you see how they have glommed on, right, or aggregated in that location. Good, so they kind of covered up the hole, right? Covered up the hole. So we have a big pile of platelets. And in the process of glomming on, aggregating, covering up the hole, right? Here they're showing the secretion of that TXA2, right, thromboxane A2. So we see that here. And the thromboxane would influence, yeah, I see. Another thing they didn't put in here was the smooth muscle, right? So the smooth muscle would be kind of out here, right, on that blood vessel. We would have smooth muscle kind of, right, encircling this whole entire structure. Like, again, we'll fix it in our version. But we would have a layer of smooth muscle, like out here, and that thromboxane A2 would tell that smooth muscle to contract, and we get our vasoconstriction right here, right? Kind of slow things down, slow down, right? Let's slow down. All right, so far we've got that happening right here, where these platelets are. Shh, slow down. Let's look over here. We see an old friend, nitric oxide, right, being secreted by endothelial cells, which we know happens, right? So we already know at least that much. We also know that nitric oxide is a vasodilator. We remember that from our big scary chart, right? That one, the local controls. It was a vasodilator. That seems strange, right? That we would have this vasodilator being secreted seems, I mean, it's like right next to this location where we've got vasoconstriction happening, right? I thought the whole idea was we wanted vasoconstriction, right? Like slow down, don't like spurt out the blood. Wow, that's weird. That's weird. Hang on to that idea for a second. Because we have this other item too that endothelial cells are going to produce. It's called prostacyclin. I guess that was the older name. But prostaglandin I2 is sort of the newer name, so PGI2. But prostacyclin is the original name. Either way is correct if you hear either one. But this substance as well is going to be a vasodilator. Vasodilator. So we've got these endothelial cells producing and secreting two local vasodilators. And we're trying to vasoconstrict. This is kind of, I don't know, it's like counterintuitive, right? Let's see one other thing. Both of these items also are inhibitors of platelets. Whoa, I thought the whole idea was we were trying to make a platelet plug, right? Why? Why do we have these things that are doing opposite things? Oh, go ahead.
It's like the modulators, so two different systems working at the same time. So one is kind of producing the platelet, that's like the overarching, and then we have the monitoring cells which are around it to make sure it doesn't get out of control.
Yeah, wow, that was so good. But now I don't have to give a lecture. That was great. That's awesome. So we have these two items that are kind of like modulators of this process. And we've seen in the body already, and this is a great additional example of, yeah, we want to do this one thing, but we don't want it to get out of control, right? So we have these controls, right? These modulators to be sure. Let's look at the locations, right, of where this is happening, right? So if we have our plug right here and that TXA2, right, like working right there at the platelet plug location. Notice how the nitric oxide and the prostacyclin, and also over here on the other side, nitric oxide and prostacyclin, see how those are being secreted by the intact endothelial cells, right? These are the not cut ones, right? Not cut. And these, whoops, they got cut, right? And now the platelet cells are aggregating over there. So, let's say the blood is flowing this way, right? Let's just say blood flowing that way, and we have blood flow, especially if we have vasodilation in this specific location, we can get, a little more blood to this location. You know what we can deliver more of if we increase the flow? We can deliver more platelets, right? Get them to glom on, but if we vasoconstrict as they're aggregating, we can slow down, allow them to like, all right, yeah, attach on, slow down, and then whatever blood kind of squeezes through, we vasodilate again, hurry up, move along, right? Move along, so that we don't see these like platelets that didn't glom on, right? So that these aren't going to start like hanging around and aggregating over here where we don't need them to aggregate, right? We want them to like, right? Move along, citizens. Nothing's happening here, right? All right, so that's the setup. It's good, right? So smart. All right. So now we've made our platelet plug, right? So we vasoconstricted. We had to work some vasodilation sort of around on either side of the vasoconstriction. to really, really get it to work like smoothly, right? And we made our platelet plug. So we've knocked out 2 things in that list for hemostasis. Our third thing on the list was clotting, right? Clotting. So our fancy word for clotting is coagulation. Coagulation, so you see that on the top. And let's look at this little chart for coagulation. Oh, it's kind of yucky. We've got vessel damage here, and of course the blood gets exposed to the collagen underneath. And we have all the stuff that we already saw, right? Happen, and we get our platelet plug. So, meanwhile, we're going to start the process of making a blood clot, which is sort of a bigger, more substantial item. The platelet plug is extremely temporary fix, right? Clot is not the ultimate goal. The ultimate goal is you want to heal the endothelium with nice, brand new endothelial cells and make it all smooth as if nothing happened, right? That's, of course, the ultimate goal. But the endothelial cells, they need time to do that. So to buy ourselves some time, we build the clot, which is kind of like, the scab, right, that would be on your skin. So this is kind of like, the scab on the inside of a blood vessel, right? The scab, and eventually the scab's going to fall off, right? We don't really want it to fall off because then it might get stuck somewhere, so we're going to have a process to melt it, right, dissolve it instead of just letting it, fall off or you scratch it off or something like that, right? Look at the face, this is so funny. But we'll have a whole process for that. Okay, so let's see. So let's build the clot first, right? We'll build like the scab, right? All right. So because the blood is exposed to the collagen, right? Besides doing the whole platelet plug thing, we're also going to trigger a cascade. We've seen cascades before, right? Enzyme cascades where an enzyme is activated, and that does something, then that does something, and that does something. So we've got a cascade here. Boy, luckily they left out the specifics, right, of the cascade. So we're going to go with that. That's good. Be grateful. Let's just focus on the end of the cascade. The end of the cascade is going to be this pre-enzyme called prothrombin, and that will be converted after cascaded to the active enzyme thrombin. Active enzyme thrombin. So of course, enzyme, we know, right? Facilitates some kind of chemical reaction, right? So thrombin is going to facilitate 2 rather simultaneous reactions. And let's look at this one. So basically on this slide, focus on the things with the asterisks, the double asterisk. So one reaction is going to be taking fibrinogen, which we mentioned before, right? We mentioned fibrinogen as one of the plasma proteins floating around. Remember? All right. So fibrinogen was floating around in the blood anyway. Protein floating around. And convert that into fibrin, specifically something called loose fibrin. Loose fibrin, which is the version of this protein that is going to be functional, right? That's going to do a job. All right. So we've got our loose fibrin, and we need to, or the body needs to convert loose fibrin into its most stable form called stabilized fibrin. So it's really our goal, right, in this whole entire thing is to end up with this protein called stabilized fibrin. That's our goal, right? But we go through a whole series of steps to get that stuff, right? To get that stuff. The other thing that thrombin is going to do is facilitate the reaction that activates this cofactor that does that final step. I took away, I didn't put in any double asterisks for here, because once we get into here, you're going to see there's a slide coming up. We open up a huge, extremely bad Pandora's box. Very bad. So I did not put the double asterisk there. I just wanted to focus on things that had names that we have a chance of remembering. Right? All right. So we have a chance of remembering prothrombin and thrombin. Like those are words. Those are decent words that we could hopefully remember. And then fibrinogen, loose fibrin, and stabilized fibrin. I hope those are words, right, that you can sort of, I don't know, relate to or something and memorize. All right. And here we've got another positive feedback loop that we see here. They could have done the dot. I think the dotted line would have been more consistent with their previous slides for feedback loops. All right. And then I think we have a picture. Yeah. So we have a picture of a blood clot, right? Blood clot. So we see red blood cells tangled up in our strands of stabilized fibrin. So that's our goal, is to end up with these strands that are capable of trapping red blood cells and making that clot, right? Like our scab for the inside of a blood vessel. Good. So, can you see, like the official name, thrombus is our official name for a blood clot. Good? Thrombus. So that kind of sounds like thrombin, right? Prothrombin, thrombin, thrombus. Good. All right. So this was the Pandora's box that I was referring to. Like, oh, once we go into that one area, then it gets really, really ugly. So this is the thing that I do not want you guys to worry about. Oh, brother, for the exam. This exam's showing up on Thursday, by the way, right? The blood one. So, oh, don't go there on this one. The only thing we're going to know from this slide, let's start at the bottom, like something we know, right? Prothrombin and thrombin. And we see like, oh, well, there's an arrow getting into there. So this is sort of where these are actually two separate pathways, two separate enzymatic pathways that can feed into this step, right, from the previous slide. And let's know the names of the two pathways. One is called the intrinsic pathway, and one is called the extrinsic pathway. Again, just know the names of the pathways. Let's know that, yeah, there are these two pathways, right, that can feed into this conversion down here that we saw in the previous slide. Good? We can fit these in. Let's see. The other thing that I want you guys to know is just that it's the extrinsic pathway that the body happens to use more, right? Extrinsic more than intrinsic. That's all I was going to say. Good? For that. So, be ready. Let's take a little side trip and look at the role that the liver plays in this whole blood clotting process. Liver, right? So far, liver was in charge of as far as blood is concerned. What was liver in charge of so far? Bile production, and we did have something from blood that was going to end up in the bile, right? So what was that? The heme, right? Heme, biliverdin, bilirubin, ending up in bile. So that's good. I don't know. Any other? What did you say? Yeah, like take out the bad ones or the old ones, right? And go ahead and start the recycling process. Okay, so far that's what we've seen. Oh my gosh, let's see. It turns out liver's going to do some stuff that has to do with clotting as well. Oh brother. So we saw stuff about bile, right? And we just reviewed that. So stuff about bile. Okay, bile we know, hopefully from anatomy, at least a little bit, that bile gets dumped, right, into, I don't know, let's review from anatomy. Where does it get dumped into? Bile. I don't know, let's see if we remember. Duodenum. Nice. All right, we'll do that more when we get to digestive. That's fine. Just wanted to see. So anyway, okay, bile dumped into duodenum. Good. What are bile salts for? I mean, we'll do it when we get to digestive. I just want to see. Anybody remember bile salts? No. That's all right. We'll do it. Huh?
I don't learn about those.
That's all right. Because I know anatomy that's kind of crossing over to physiology. All right, so we'll talk. That's all right. No big deal. No big deal. But anyway, dumped into duodenum is going to be for fats, but we'll talk about all the details later. So anyway, okay, along that small intestine. Bile salts are going to help us to absorb this particular vitamin, yet another vitamin that we're adding to our list of the vitamins from the list that we're really going to learn in detail, right? So now we've got vitamin K. Vitamin K. Does anyone remember from the original list of vitamins, what category of vitamins K was in? Oh my goodness, who said that? Oh my god, that was like no pause. Awesome. Say it again. Fat soluble. Whoa. Oh man. The reason I had you guys memorize like this, this one, this, like these specific sections in that table. Okay, this is why. So that on this day, boom, like Dina can say, oh, fat soluble. Right? So good. That's awesome. Okay, I'm glad. So, all right, so vitamin K was one of our fat-soluble vitamins. I just happened to mention, we'll talk more on digestive system, that bile salts help us to absorb fats that we've eaten. All those details are going to come up later in digestive. But if we've got a fat-soluble vitamin that we need to absorb, well, let's do that at the same time, right? Using bile salts while we're absorbing fat in the diet, right? That works. It works together. So we're going to absorb vitamin K in that small intestine. It's going to end up in the bloodstream, of course, right? Just like everything else. And let's cycle back to the liver because we know. Oh, you guys have to know this from anatomy. You have to. Let's review this. What is the specific part of the vascular system that is in charge of delivering the blood from the digestive system to the liver? Please get this one. Oh my gosh, hepatic portal system! Ah, yay! Okay, so hepatic portal system. Woo! Awesome. So hepatic portal system in charge of gathering, right? All of that blood from a small intestine, especially, right? Small intestine, but also other parts, and delivering that first to the liver, because what is the liver going to do first before we send it to the heart? We'll talk more on digestive, but what is, like in general, liver going to do? I know we mentioned it in anatomy. Super general. Like filter, right? Cleanse, right? We just took in a whole bunch of stuff from God knows where, right? Just took a whole bunch of stuff in. And it would be super smart to cleanse before actually delivering it to the heart, and then we send it all over the place, right? All right. You know, you never know what's in there, right? In the food. Pesticides, bacteria, right? Oh my goodness. So, all right. So we sent it to the liver. You know what? While we're in the liver, let's drop off that vitamin K that we took in, right? And that's where we're going to use the vitamin K to make a lot of the clotting factors that we just saw on the scary slide, right? That I didn't have you guys memorize, but they're there, right? They're there. So, cool. And then we can do all the rest of the stuff, right? And end up with our prothrombin and thrombin. Good? All right. Vitamin K. Fat soluble. Super awesome. All right, so we made our blood clot. And, let's see. Like I hinted at before, we do not want the blood clot to be permanent, right? We want ultimately the endothelial cells to heal themselves, right? Regenerate and make a nice new smooth endothelium underneath the clot. and then we can get rid of the clot. Just like when you have a scab on your skin, underneath the scab, the skin is regenerating itself, and then eventually the scab falls off, and then you have nice new skin, hopefully with not a scar or something like that. Hopefully you have nice new skin. But in the bloodstream, we do not want a clot to just fall off, because what's going to happen as it travels through the bloodstream? It's going to get stuck somewhere, right? And if it gets stuck, well, I mean, anywhere it gets stuck is not going to be good, but if in particular it gets stuck in the coronary circulation, we cut off the blood supply to heart muscle. What's that? That's a myocardial infarction or heart attack. Okay, so that's not good. If the blood clot gets stuck in one of the blood vessels in the brain, stroke, right? We cut off the blood supply to a section of the brain, stroke, no good. If the clot gets stuck in maybe the blood vessels in the lungs, pulmonary embolism, right? Gets stuck, cut off blood supply to a chunk of lung, no good. All right, so it's bad situation. So we have built into the body a way to dissolve, right? Dissolve such things. We also have a way to control the clot so that it doesn't, just like the platelet plug, we had control so the platelet plug doesn't become too huge. We have ways to, keep the clot like just the right size so it doesn't become too huge. And then eventually we're going to dissolve the whole thing. All right, so we'll call that anticoagulation, right? Or here they have anticoagulant mechanism, but you can say anticoagulation. I think that's easier. Anticoagulation. Great. Look at what we've got here. Well, here's endothelial cell. Here's something to control thrombin. That should look familiar, right? Thrombin should look extremely familiar. It's a couple of slides back, right? And it was a couple of slides back for making the clot, right? Right? Okay. Let's see where we're going with this. Thrombin looks like they actually gave us a little specific shape in there for this item, protein C. Protein C looks like they fit together, right? They did that on purpose. So it looks like thrombin can attach to this particular protein also, floating around in the area, protein C, and it activates this protein C. And this activated version of protein C acts upon these two things are from scary slide, right? Scary slide. And you see the negative signs? So activated protein C acts upon two items from those scary pathways, right? And turns them off. So if we're going to turn off items in the pathways, right? Items in the pathways that were leading ultimately to clotting, what are we doing? We're turning it off, right? We're turning off like the whole process, right? Okay, so this is how we can stop the clot, right? Or you can maybe think of it as start turning it off, right? Okay, but what have we used to do it? Thrombin. What's with thrombin, man? Right? It's like making the clot, it's like stopping the clot. It's a double engine, right? Huh. Turns out, thrombin kind of is a double agent, right? That's cool, right? To be a double, isn't it cool to be a double agent? I don't know. Is it? I'm not sure. I'm looking at Jonathan for like some kind of feedback from that. Okay. Has anybody? What is that movie? What is it? John Wick? Is this the one we were making fun of? We haven't seen it. Has anybody seen it yet? It just opened, right, this weekend? Didn't it?
Got Keanu Reeves in it.
With our friend?
Yeah, we're not gonna watch it.
Yeah, I know. You guys aren't gonna watch it. It's too funny. I feel bad now.
Yeah.
So bad. I was being encouraged to let it go, right? Let it go. Man, I'm not gonna break out into song either. Let it go song. But I was being encouraged to let that go and say I'm giving Keanu a chance. It's just the trailer doesn't look that good. Keanu or no Keanu? The trailer just looks a little bit like, I don't know. All he's doing is like shooting guys point blank, you know? And it's like,
So like be ready for him.
a little bit. I like a little more intrigue, you know? Anyway, I don't even know if he's a double agent in that thing. It just popped into my head because we were talking about double agents. Anyway, there's Thrombin. What was that one with the double agents? It was that Taylor Tinker thing. Almighty. That movie was like the most confusing movie I think I've ever seen. It's so confusing I can't remember the name of it properly. Yes, that one. They were all double agents in that movie. And I did not understand whatsoever. So anyway, those are this thrombin, double agent thrombin, right? Look at this. Procoagulant, that means encouraging coagulation. Anticoagulant, stopping coagulation. So thrombin can do both, and it does do both. So we've already seen where thrombin works to convert that fibrinogen to fibrin, right? Remember, loose and then stabilized fibrin, and that was going to tangle up all of our red blood cells to make the clot, right? All right. Also activates some of the factors in the scary pathways and stimulates the platelets, right? In the sort of beginning when we were making the platelet plug in the first place. So these are things that thrombin is in charge of encouraging coagulation. And then the thing that we just saw was activating that protein C, and then activated protein C was in turn in charge of stopping some of those items from the two scary pathways, right? So we're going to stop coagulation. Huh, that's interesting. Why would we do that? Have one thing that's in charge of like two opposite things. Kind of, I don't know, kind of negative feedback and kind of positive feedback, right? Sort of, right? But it's the same idea that we saw before when we we had multiple items modulating our vasoconstriction, right? That whole thing in the platelet plug, we had multiple items. So here we have this one item sort of right in the middle, right? Controlling so that we don't go overboard in either direction. Huh, right. Kind of interesting. So, we have said that the clot was like temporary, right? So now we have to, and we now have figured out a way using thrombin to control the clot so it doesn't like grow too big, right? We're going to just keep it somewhere in here, right? Like just the right size, not too big, not too small. And now let's say the endothelium has healed itself and we want to actually dissolve the clot completely, right? So we're done kind of maintaining it. We've let the endothelium heal itself, and now we're going to dissolve the whole thing, let it just, right? All the small pieces fizzle away, no harm done, right? So, this is the pathway we're going to use to end up with soluble fibrin, right? Stuff that can just melt, right? Dissolve in the plasma and just float away. All right. What do we recognize? We recognize plasminogen. We've mentioned it, right? Yes. So we recognize plasminogen. We need to activate that and convert it to plasmin, which in turn will break down the fibrin, right? This fibrin here, we could also call that the stabilized fibrin, right? Remember we had that at the end of the other slide, stabilized fibrin. So that's where we're kind of picking up and breaking down that stabilized fibrin into soluble fibrin. Good? All right, so that's kind of straightforward. Let's look at the things that activate that plasminogen into plasmin. Proteins that are secreted by those endothelial cells. Does that totally make sense? We just healed the endothelium, right? So when the endothelial cells are back to being intact, woo! send out these guys, right? And they'll go ahead and activate dissolve the clot. Good. Good? It's very nice. It's very elegant, this whole process. So, here we've just got a table. Let's see, this is a table just summarizing endothelial cells, right? So you can kind of go through that. This is a lot of stuff that we already talked about. There might be a few things in here that are slightly more detailed than, than we talked about. Just go with what we talked about. Okay? But this is just to see it in one place, if it helps you. If it doesn't, but if it helps you, then go with it. All right, so that is the end of the blood section. Good. So let's take our break, and then when we come back, let's do a bunch of housekeeping first, right? Before we move on to anything else. Good? Including like setting ourselves up for the group presentations, because I want to do that on a day this is like a relatively light day compared to what we're looking at on Thursday. So let's also set up our group presentations. Good. So maybe when you come back, start thinking about who do you want to work with, right, on the group presentations. Good? All right, so let's come back, I don't know, like 9:30, 9:35, somewhere in there.